Okay, good afternoon, ladies and gentlemen. The webinar is going to start immediately. To all participants, please use the virtual background and fill the attendance form from the link provided by the committee in the chat box. We will start the session. The Honorable Puji Lestari SP, FSI, PhD, Chairman of Research Organization for Agriculture and Food, ROAF National Research and Innovation Agency BRIN Indonesia, Head of Research Center in Research Organization for Agriculture and Food BRIN, participants from National Gene Bank Rice Research Center Laos, participants from Mardi, Malaysia, speakers, moderator, and all researchers in BRIN. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. It is a great pleasure to welcome you to the ROAFERIAN 7th series, held by Research Organization for Agriculture and Food, National Research and Innovation Agency BRIN, Monday, July 29, 2024. Today, we are going to engage a very interesting topic about leveraging rice germplasm resources for breeding success. Ladies and gentlemen, next is welcome remark from Chairman of Research Organization for Agriculture and Food BRIN to Mrs. Puji Lestari, SP, MSc, PhD. Time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. First of all, I would like to thank God, Allah Subhanahu wa Ta'ala, who has been giving us blessing and mercy, so we could attend today's event. And it is my great pleasure to welcome you to today's webinar series of 7 ROAFERIAN organized by ROAF, Research Organization for Agriculture and Food BRIN, with today's theme of leveraging rice germplasm resources for breeding success. I would like to extend a warm welcome to all head of the centers under ROAF who are joining this webinar, and our esteemed speakers, Dr. Gaukham Philay Hyam from Nafri, Laos, Ms. Rahinisa Kamaruzaman from Mardi, Malaysia. I'm very happy to have you both today. After 5 years ago, we ended our research project on BSF, AFOO, even on the rice project. And I also give high appreciation for the research speakers, Dr. Dani Satyawan from this Organization for Life Science and Environment, BRIN, Dr. Aris Hermanses from this Organization for Agriculture and Food, BRIN, who have agreed to share your expertise and insight with us, or your contributions are highly valued. My gratitude is also addressed to moderator and the head of the Center for Food Crops, Dr. Yusdira Nugraha. I acknowledge our colleagues from Mardi and Nafri probably joining this webinar, and thank all researchers and all participants joining this webinar. Ladies and gentlemen, why we are organizing today's webinar, which focus on rice breeding with its linkage to the genetic resources. As we know that the increasing challenge of climate change, pest and disease pressure, and the need for higher yield to feed a growing population is urgent to continuous improvement in rice varieties. Rice germplasm basically contribute a unique genetic trait that can be harnessed for breeding. Furthermore, a rice gene bank is an essential resource for the conservation and utilization of rice genetic diversity, playing a critical role in the sustainability and advancement in rice breeding programs. They maintain a wide spectrum of genetic resources, and these rice gene banks provide breeders with the raw materials needed to respond to evolving agricultural challenges for increased food production. Additionally, rice gene banks are a cornerstone of modern breeding effort, enabling the continuous improvement and adaptation of rice varieties to meet the demand of the future. Those they facilitate international collaboration and exchange of genetic material, promoting global security and agricultural sustainability. while advanced breeding strategies such as marker city selection, genomic selection, genome editing, along with the cost-effective approach to high-throughput genotyping and phenotyping, together enable breeders to effectively leverage rice germplasm resources, leading to the development of supervised varieties that can meet future agricultural challenges. Therefore, international cooperation in rice breeding acceleration is of paramount importance as it fosters the exchange of knowledge, resources, and technologies, thereby enhancing the global effort to improve rice varieties as now we are supporting by this webinar today. Collaborative effort among countries, research institutions, and international organizations enabling the sharing of diverse rice germplasm, which broaden the genetic base available to breeders and accelerate the development of resilient and high-yielding rice varieties. This cooperation also facilitates the pooling of expertise and technological advancement, which can be leveraged more effectively through joint research initiative and sharing fund. Ladies and gentlemen, Research Organization of Agriculture and Food, BRIN, is highly concerned about rice breeding to support food security in the country. Therefore, collaboration research to boost the achievement of goals on this research and innovation are in the discussion by BRIN and institutions from Laos and Malaysia. Ultimately, international cooperation in rice breeding not only accelerates the pace of scientific discovery and varietal development, but also enhance the resilience and sustainability of rice production on global scale. Thus, today's webinar on leveraging rice germplasm resources for breeding success aims to bring together experts, researchers, and stakeholders in the field of rice breeding to discuss the strategic utilization of rice genetic resources. The specific objective are to share knowledge of how to enhance rice breeding for food security, including germplasm collection, advanced technology, and breeding strategy. Share success stories and discuss some of many concepts of the joint research project among parties probably come up during discussion. And to strengthen networking and initiate collaboration between breeding researchers and national institutions from other countries, especially from Malaysia and Laos. This is all of my remark, and I apologize not to fully join this webinar since I also have offline meeting in international event. Now I am in Sumedang in Pajajaran University, but I'm very sure this international webinar will be profitable and fruitful for everyone present here, and continue our collaboration between BRIN and Mari and NAFRI. Also other stakeholders will fulfill this webinar with the concrete implementation of any research and innovation project. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Puji, for the speech. And to all participants, before we start the discussion, we will have a photo session first. Please activate your camera and for the committee to take the screenshot. I will start count from first slide. Ready? 1, 2, 3. Second slide. 1, 2, 3. And third slide. 1, 2, 3. Thank you. Ladies and gentlemen, now we are going to start our discussion for today, which will be led by Dr. Yudhistira Nugraha, SPMP, Head of Research Center for Food and Crops, ROAF BRIN. Before that, I will read his CV first. Dr. Yudhistira Nugraha, SPMP. His area of expertise is in rice breeding and genetics, physiology of rice plant. He got his bachelor degree in plant breeding at Jenderal Sudirman University, Indonesia in 1999, master degree in plant breeding at Pajajaran University, Indonesia in 2010, and PhD in plant breeding and biotechnology at IPB University, Indonesia in 2016. He is currently head of Research Center for Food Crops BRIN. Previously, he worked at Ministry of Agriculture. He has released hybrid rice varieties, irrigated rice varieties, and swampy rice varieties. And his recent publications are Deciphering Genetic by Environment Interaction, Pests and Diseases Interaction, Grain Quality and Response to Fertilizer for Targeting Height, Advances in the development of rice varieties with better nutritional quality in Indonesia, and cover change and food security in Central Sumba, and bioactive compound profile and their biological activities of indigenous black rice from Java and East Nusa Tenggara. So without any further ado, to Dr. Yudhistira, time is yours.
Thank you very much, Ibu Andulusi. Do my sound is clear for you? Is my
clear.
Okay. Thank you, Ibu Andulusi. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. First of all, we praise to Allah Subhanahu wa Ta'ala, the God Almighty, for the wealth and time that given to us. And without His blessing and mercy, we can gathering together in this special occasion, the 7th ROAFERIAN of Research and Organization for Agriculture and Food. The Honorable and the Chairman of Research and Organization for Agriculture and Food, Dr. Puji Lestari, the distinguished invited speaker, Dr. Aris Hermansyah, a researcher from Research Center for Food Crop BRIN Indonesia, Dr. Dani Setiawan, researcher from Research Center for Genetic Engineering BRIN Indonesia, Dr. Kokam Hali Hung, from National Gene Bank Research Centers, NAFRI, Lao, PDR, and Niza Kamaruzaman from Research Center Mardi, Malaysia. And also for the audience of this webinar, I see some researcher, academician, extension, and all participants who concerned in agriculture. In this great occasion, we are going to discuss and sharing the important topic, which is leveraging rice germplasm resource for breeding success. These topics become main issue in Indonesia and elsewhere, who most people consume rice as a staple food. Today, the price of food is going to spike, and in the same time also we are facing climate change. We need to develop a rice variety that can cope the biotic and abiotic stresses, which become more severe from now on and in the future. So we have 4 speakers who will talk about with related subject and on their expertise, and of course from different countries, from Indonesia, from Malaysia, and from Laos, who also have different breeding system in each country. So it will be interesting to discuss. And for the speaker, I will give the rule of thumb of this session. So each speaker have 20-minute allocation time for deliver his presentation, and after it will continue parallel, begin with Dr. Aris, Dr. Dani, Dr. Kaikam, and Mrs. Rahiniza. So, if the speaker, I will consult the speaker if approaching 2 minutes before allocated time, and the Q&A will be held after all speaker presented the presentation. Without further ado, I will call Dr. Aris Hermansyh. So before Dr. Aris Hermansyh present his talk, I will read first the CV of Dr. Aris Hermansyh. So please, the CV Dr. Aris Hermansyh. Dr. Aris Hermansyh, his expertise is in area of genetic and plant breeding. He got a bachelor degree agronomy from Brawijaya University in 2021. He got master degree in IPB University in 2025, and he had a PhD degree from University of Adelaide in 2014. His career, he's the senior researcher in Research Center for Food Crop BRIN. And before that, he's a researcher from Ministry of Agriculture. And most of his career in rice breeding. He have released some rice variety, hybrid rice variety, irrigated rice varieties, upland rice variety, and swampy rice variety. He also published some publication related to the rice breeding. So, Pak Aris, you have 20 minutes. Time is yours.
All right. Thank you, Pak Yudis. I hope everyone can hear my voice clearly. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to thank the organizer of ROAFERIAN 7 for giving me opportunity to share our recent activity in our research group under Research Center for Food Crops. I just want to check, should I present with myself or from the organizer, the PowerPoint? Pak Yudis, should
Pak Aris, you will present your PPT or by the organizer?
if I can share with my computer then.
So, please, Pak Aris, you can do that.
All right. Hope you can see my slide now. Right. In this time, we will share about the current status of rice breeding in Indonesia. I'm from the rice breeding research group under the Research Center for Food Crops in BRIN. And of course, because there are many research centers in Indonesia who work with rice breeding. So what we will share in this presentation, mainly from our research group mainly what we have done in our research group. Before we talk about rice breeding in Indonesia, maybe it's interesting to see some of rice facts in Indonesia. As we know that Indonesia is the 4th largest rice producer and also consumer in Indonesia in the world, after China, India, and Bangladesh, I think. And every year, rice grown in more than 10 million hectares. It's very large area, of course. And why it's so important? Because it provides almost 40% of Indonesian people calories and about 30% of protein for Indonesian. And then, interestingly, rice in Indonesia is grown in very diverse area, from the hill up to 1,200 meters above sea level, up to the coastal area. You see in this picture, in the top picture, there's a rice field in Indramayu where farmers grow rice very near to the sea in the coastal area. And in the bottom picture, I took this picture several years ago when we have rice breeding or rice field trial in Simalungun, North Sumatra. It's about 1,100 meters above sea level where farmers still grow rice in that area. And total about 531 varieties of rice varieties already released in Indonesia for commercial cultivation now. Rice in Indonesia mainly grow in irrigated area, and about 65% of rice produced in irrigated lowland area, and about 30% grown or harvested from the rain-fed area, and small portion harvested from the swampy area. This includes tidal swamp or inland swamp. And this is data from the 2022, yeah. The rice productivity in Indonesia quite variable. In Java, in under irrigated lowland area, farmers can get more than 5 tons per hectare. But in some area of Kalimantan, in Papua and some area of Sumatra, even in lowland area, in irrigated lowland area, farmers only can get as high as 3 tons per hectare. And this also happen under the upland condition. Upland area in Java or in some area of Sumatra and Sulawesi, farmers can get up to 4 tons per hectare rice. But then in some area, the productivity is very low, even under 2 tons per hectare. Depend on the rice ecosystem, the constraint for rice production are different. In irrigated area, a major problem are brown plant hopper and bacterial leaf blight, also blast and tungro. So mostly the problem for irrigated area is related with the biotic stresses. In the rain-fed area, drought and blast is the 2 major problem, besides weeds and also brown plant hopper. While in upland, of course, drought is a main problem. In some upland area with acid soil, aluminum toxicity becomes a problem. And also a blast also become a main problem, and also phosphorus deficiency. While for swampy area, the main problem related with the iron toxicity, there's submergence and salinity in some area. And for the biotic stress, blast and tungro are the major problem in this area. And related to that problem, rice breeding objective in Indonesia mainly targeted for yield potential. And then we aim to increase the abiotic stress tolerance, pest and disease resistance. And we also now give some attention on nutrient efficiency, especially for nitrogen and phosphorus. And not last but not least is nutrition values. So this is also important objective in our rice breeding program, because in Indonesia we have some problem with malnutrition. So we try to improve our rice varieties for macronutrient content like zinc and iron. Also, we try to develop some rice varieties with low glycemic index. When we want to release rice variety in Indonesia, there are some minimum requirement for rice to be released in Indonesia. And of course, this should be taken note by breeders when they want to release varieties in Indonesia. For example, for irrigated lowland, it has to have yield potential at minimum 9 tons per hectare, and it must be resistant to brown plant hopper and leaf blight. While for swampy area, the variety released for this area must have yield potential at least 6 tons per hectare, and it must have blast resistant, because this is the main disease in swampy area, and it also have to have iron toxicity tolerant. While for upland, the minimum requirement is 7 tons per hectare with blast resistant. And differently with hybrid rice, our government required the hybrid rice which released in Indonesia, at minimum, they must have yield 11 tons per hectare. And similar to irrigated lowland, it has to be resistant to BPH and BLB. And another requirement is the F1 seed production must be efficient. At least the parent should be produce at least 1.5 tons per hectare of F1 seed. How is the variety release in Indonesia at the moment? So our data show that since 1940 until this year, the total is 123 rice varieties already released in Indonesia, and mainly released as inbred, about 416, and 107 of them are hybrid rice. And among the variety released, currently about 155 varieties which are grown by farmers. This is data from the Directorate of Food Crop Seed. And among the ecosystem, most of the rice varieties released for lowland area, almost more than 300 varieties, and some for swampy and upland area. And if we see the variety adoption in the last year, we found that the top 3 varieties at the moment is in Pari 32, Ciherang, and Mekonga, which cover almost 50% of rice area. And it's interesting that in Pari 32, it's released in 2013, and it replaced Ciherang as the first variety in Asia in 2021. So it took about maybe 7 or 8 years from this new variety to become dominant in Indonesia. Now, I just want to share you some of our rice breeding activities in the Research Organization for Agriculture and Food under the BRIN. We have quite diverse rice breeding activities now in our research center. Some researcher or there is a team who work with multi-tolerant abiotic stress, and then we have researchers who work with improvement of virus resistance, blast resistance, and our team also work with hybrid rice, biofortification. We now also started to work to improve or to reduce low glycemic index in rice. And also we have some work on nitrogen efficiency. And with the support from the local government, we also do some local variety improvement. This is our work on the development of multi-tolerant rice varieties. So Ms. Trias Sitasmi and also with Pak Yudi's team try to develop varieties which combine more than one stress tolerant. In this case, they want to combine submergence, salinity, and drought into one varieties. This is, of course, one of the adaptation strategy for climate change. And now they have yield trial in target area for submergence, salinity, and drought. We hope that we can release our new varieties in next year or in the next 2 years. And for this research, we have collaboration with the Ministry of Agriculture, with the BSIP in Sukamandi, and also with some seed industry. Regarding the biotic stress, we now try to develop virus disease resistant using new technique, genome editing. We have in Indonesia, we have quite serious problem with the rice virus, which is transmitted by the brown plant hopper. And unfortunately, there is no genetic resources for this disease. So we try to use genome editing technique to develop virus resistant variety. And this research led by Dr. Nanas and Dr. Mitsi, also with Pak Firmansyah, who expert in virus breeding. Another topic is on the blast disease resistance. Blast, we know that blast is a major disease both in upland and also in lowland area. And our researcher, Ibu Siti Yuria, and team, she tried to introduce some of blast resistant genes from the wild species like Oryza glaberrima and Oryza rubipogon into our commercial varieties. And I believe Bu Siti Yuria now has some promising lines under the field trial, and she tried the line in Bogor here, also in Papua. So it's interesting to see in next few years to get new varieties released from this project. And then another project which run in our group is for the biofortification. This work led by Dr. Untung Susanto. He and team try to improve the zinc and iron content in rice grain, and he used quite diverse technique, genome editing, and also using conventional breeding, and also using mutation breeding. And we hope that we can get rice varieties with higher zinc and iron content compared to the existing variety like in Pari Nutrizing. And for this project, Pak Untung has collaboration with Harvest Plus, Ministry of Agriculture, and also some work for the physiological work with Tokyo University and with the Universitas Jenderal Sudirman. Under collaboration with JIRCAS, Japan International Research Center for Agricultural Sciences, we also try to develop a nitrogen efficient rice. We know that nitrogen is the most important fertilizer in rice, and the price also continuously increase. And for the environmental view, the use of excess nitrogen also not sustainable. So we try to develop new rice varieties by introducing some KTL responsible for the nitrogen transport, like KRR61 and OS-ACTPK1. And we introduced this into popular rice variety Ciherang. And now this work under field trial, and we also hope that we can have a new variety with more efficient to nitrogen fertilizer. And Bu Swishy Magat and Pak Bayu work on this breeding now. For the hybrid rice breeding, most of all of the rice varieties that all of the hybrid rice varieties which released in Indonesia use 3-line system. It means we use CMS, cytoplasmic male sterile, and maintenance to produce the female parent, and we use Restora as male parent to produce F1 seeds. And in our group now we try to develop new hybrid rice using 2-line system. It means we need female parent based on the thermosensitive genic male sterility. And we try to use genome editing to develop new TGMS line. Also, we also introduce some source of TGMS line from IRI to support this breeding program. And this work led by Bu Indra Stuti, and now Bu Yuni Widya Stuti doing her PhD degree in IPB for this research project. And then with the support from the local government of Riau, one of our researcher, Pak Palin Sinaga, tried to improve the local varieties in Riau. Riau is known as very rich with the local varieties, and the local government is quite support for improvement of these local varieties. And Pak Palin and his team already registered some local varieties from Riau, and he also doing some crossing and selection using this material to get new line which more adapted to Riau. I think that's all for the breeding program that we have in our group now. As a summary, I just want to highlight that research priorities in the breeding program mainly still to improve the yield potential, and then to improve biotic and abiotic stresses, fertilizer efficiency, and nutrition values. We also has already incorporated new breeding technique as genome editing, and also use some advanced phenotypic platform in our breeding program. And then lastly, I would like to give emphasis that for the future, we want to give emphasis on the co-development of new breeding lines between our research center with the private companies or local government. We think that it's very important to accelerate the variety adoption of rice, which we develop in the future. I think that's all from me, Pak Yudis. We hope that the information will be important for other people. And thank you.
Thank you so much, Pak Aris. You have saved about 2 minutes from the allocated time. And what we got from Pak Aris' presentation, I think there is what do you call it? Rice breeding program in Indonesia have been released about more than 500 varieties, and maybe in the future, BRIN also will release some rice varieties. For example, for multi-stress tolerance, hybrid rice, and for virus resistant of rice and others. So all of those work have been under progress in the research group of Pak Aris and under pipeline to be released. Thank you very much, Pak Aris. Next speaker will be Dr. Dani Setiawan. Before Dr. Dani Setiawan presented his talk, please show me to. I will read the CV of Pak Dani. Please, okay. So Dr. Dani Setiawan is a researcher from Research Center for Genetic Engineering, or ROAFERIAN BRIN Indonesia. His expertise is in genetic moleculars and molecular biology. He got a degree for bachelor degree from University of Queensland, Australia, and he got master degree from Biotech Institute Technology Bandung, Indonesia in 2020, and he got PhD from Seoul National University, Korea, and he's now in as a researcher in Research Center for Genetic Engineering BRIN, and before he's a researcher from Ministry of Agriculture, same with Pak Aris, and many publications has been published by Pak Dani and team. And without further ado, Pak Dani, you have 20 minutes for your presentation. Time is yours.
Thank you, Pak Kapus. Allow me to share my screen. I hope everyone can see my slides. Okay, good afternoon, everyone. So without further ado, I think I will just first try to limit the scope of genomic data for this talk. So what I mean by genomic data is basically just any data resulted from utilization of genomic information. So it's not necessarily limited to whole genome sequence data, but may also involve information obtained from DNA markers. But those markers is usually chosen because you are interested in a certain position in the chromosome or vicinity to a gene of interest. And a lot of the things that I present here is not meant to be an exhaustive review of what's every techniques that's available today, but rather mostly based on previously completed work, whether it's here in BRIN or in my previous institution in the Ministry of Agriculture. And the source of genomic information is definitely also depends on your needs. So if you want to know every single base in the genome from telomere to telomere in every single chromosomes, then you may need to use sophisticated technique and equipment such as whole genome high fidelity long reads, such as PacBio Hi-Fi or double read nanopore sequencing. But if you are not really interested in areas with a repetitive sequence, then you can also use whole genome short reads, which can show the sequence variation in regions without a repetition. And if you're only interested in sequence within genes, then you can use RNA sequencing or exome sequencing. And if you are interested in just get some sample information from every chromosomal region in the genome, just to look at the trend for genetic diversity in each segment of the chromosome, then you can use even scarcer sequencing data from reduced representation sequencing, or just using DNA markers, which represent one polymorphic site for every 1,000 or million bases. And one of the ways to obtain markers is definitely by using whole genome resequencing. When you get the sequence data back, then you can just align it to the reference genome and see if there are some sequence change in some part of the genome that you're interested in between the plants that you have sequenced. And if you see polymorphism, then that's probably the one that you need to focus on. But sometimes you may not need to know every single sequence DNA in the genome. You can just use sampling techniques so that you can sequence less part of the genome, say 1% or 2% of the genome. That way you can minimize the cost of the sequencing step. And you can use a technique like genotyping by sequencing, which is basically a reduced representation sequencing around the restriction sites that you can find in the genome. So once you obtain those markers, then sometimes you want to narrow it further to some markers that you actually really interested in. And for that, sometimes you need to convert the marker data to something that you can easily detect very quickly with much lower cost using equipment that's very simple and easily available in your lab, for example. So the most common approach usually just people convert it to PCR-based marker, which is then detected using gel electrophoresis. Using that kind of approach, for single nucleotide polymorphism, you can use technique like tetra-arms or SNAP. While for insertion and deletion, you can just usually design flanking markers around the indels and then just run them on the gel. And depending on the resolution that you have, you may need to run it on just simple agarose gel, or you can use polyacrylamide gel electrophoresis if you want higher resolution. If you have sequencing machine like Sanger sequencing or capillary electrophoresis, then you can obtain even higher resolution. Then you can adapt it using technique like snapshot, which basically just add a fluorescence marker to your fluorescence stack to your empty cons, and then you can run everything together in one single electrophoresis in the capillary electrophoresis or in your Sanger sequencing. And an even faster technique is using real-time PCR-based techniques such as CAF here, where basically you just purchase a primer with a label, and then you just run it in a real-time PCR. So once you finish your PCR, you can like just know the genotype of your sample right away. So also another thing that you need to note when you try to design primer is that you should be aware that rice genome actually has many regions that have a high similarity to each other. For example, here in the end of chromosome 8 has a very high similarity to the end of chromosome 9. So if you're not careful when you are designing primers for markers or genes that located in that position, you will inadvertently also amplify a sequence from this area also. So instead of having a single PCR product, you have 2 or 3, which can confuse you later during the downstream analysis, or even worse, if you try to sequence the PCR product, then you will have a mixture of sequencing and your reads will be all messed up. So my recommendation is if you try to design DNA primers in rice, use tools that is aware of the available sequence data in the genome, like primer blast, which basically other than trying to design candidate markers for your candidate primers for your markers, it will also blast those primers to make sure that it only align to one part of the genome and it will not amplify any other part of the genome. That way you can have a very specific primer product when you do the PCR later on. And since my task today is supposed to be limited to the pre-breeding activities only, so as you can see from this diagram here in the breeding activity pipeline, I will mostly focus on the 4 activities on the left, from the crop design to the genetic resource management to the some aspect of phenotyping and also genetic analysis, which include genetic diversity complexity studies and also fetal identification, and also marker design once you obtain the marker that's most associated with the trait that you are interested in. So first, for genetic resource management, this is one case study that we did on soybean, and don't worry, this is also applicable in rice, because rice is also one of the most better characterized genomically compared to other crops. And the aim of this project was to create a soybean core collection based on genotypic data, because as you know that soybean is an introduced crop in Indonesia, so the natural genetic diversity for Indonesian soybean is not that wide, so we need to supplement it with accessions from other parts of the world to maximize the genetic diversity available in the world. So to do that, we look at USDA soybean collection, where they have already genotyped their collection using 50,000 SNPs, genotype using Illumina Infinium chip, which look like this basically. So like in every box here actually contains like 50,000 SNP markers. And for each box here, you can hybridize your DNA sample. So for each chip here, you can hybridize 24 samples at once, and then it will give you data on 50,000 SNP markers for all those 24 plants that you have. And BRIN actually has the equipment to read this kind of chips. So if you have a lot of research grant and interested in using this type of technology, you can just contact the BRIN sequencing lab to see if you can use their facility to do that. And when we look at the genotypic data from the USDA collection, you can see that in their collection, they are biased to certain family groups. So some family groups are actually enriched in their collection compared to other family groups. So if you just take a random subset of this collection, say for example 400 subset of the whole collection, then you will also maintain this bias. So you will tend to get enrichment in some of the population. So you get less diversity if you focus on some of these families rather than like maximizing the genomic diversity. So we use this utility here called Hunter, which basically just try to select the smallest number of accession with the maximum genetic diversity. So in the end, as you see here, that you get less number of accession, but the genetic distance between them is almost equal to each other, thereby maximizing the genetic diversity between each member of the core collection. And we can also use analysis such as principal component analysis from the genotypic data to help you visualizing the genetic diversity of your existing collection. For example, since we have also genotyped some of our soybean collection in our gene bank, we could see that Indonesian soybean collection is actually occupying only a very small portion of the all available diversity in the worldwide soybean collection. So if you want to make sure that you get all of the possible genetic diversity available, then you definitely need some samples from all these underrepresented regions here in this chart here. And we definitely also identified if some of the existing collection originated from overseas also can fulfill that kind of criteria, and some of them do. Some of them actually can fill some of the areas very well, but as you can see that some areas are still underrepresented, so you need to fill in this region as well if you want to complete your genetic diversity collection. And the marker data can also be used to remove duplicates, because basically there is no point in keeping more than one copy of the same materials, because it's just adding cost and labor to keep them together. And you can also analyze the marker to find like the smallest subset of markers to allow you to discriminate between every single accession that you already have, which will be useful for cheaper way to track all the progenies and all your collection in the future once you produce newly developed cultivars from your breeding program. And as I said before, similar resources like in soybean is also available in rice, and maybe even better, because IRRI actually has already sequenced the genome of 3,000 of their collection, 3,000 rice in their collection, and they share it for free in this SNP-seq database. And if you think that that's not enough, you can also consult this rice farmer database here from China, where they have additional sequence data for 1,700 rice genomes. And if you're only interested in doing the genotyping of your material that you don't know if there are some similar accession or not in IRRI collection, you can also use services that IRRI provided in the past. I think we use their service, which use a SNP chip like the soybean before, but contain 7,000 SNPs. And I think they are currently in the process of like renewing the technology here so that it can be cheaper and also much more effective, because in some cases 7,000 is simply just too much and just too expensive. So watch this space if you are interested in doing this kind of analysis in the future. And we have also utilized the sequence variation database in IRRI to develop polymorphic markers of our own when we try to develop a mapping population for our rice collection, where we try to map brown plant hopper resistance gene. When we try to cross untuk Rajab to TN1, we want to develop many more markers to map the exact location of the resistance gene. We just tried, since we don't have the sequence of both untuk Rajab and TN1, we just try to find the closest family of untuk Rajab in IRRI database and also in the TN1 family, and then look at the SNP database and see which SNPs are consistently in the same within groups but different between the two groups here. And if we obtain like about 70 to 80% polymorphism in the markers that we select, which is a big saving of money compared to if we just select the marker randomly. Okay, so once you obtain the markers, like I said before, in our case, we try to do QTL mapping. But currently, other than using the traditional linkage-based QTL mapping using a segregating population, the whole segregating population, we are also interested in exploring mapping QTL by sequencing, where basically it's using the principle of bulk segregant analysis. So for example, if you try to find the genes responsible to make rice short or tall, you can just cross a tall rice with a short rice. And then in the F2 segregating population, you choose a 10% tallest plant and 10% shortest plant, and then bulk them together and sequence them together as a bulk and along with one of the parents. So say maybe you take the taller parents here, and once you get the sequence back, then you try to find sequence difference that exists between the two bulks But then you also try to count the allele frequency of the tall parents, and ideally, if the SNP is responsible for the tallness of your population, and it should contain allele originated from the tall parents only, right?
Dr. Dani, you have 2 minutes left.
Okay, sure. Okay, so basically that's what you do, and then you plot off this allele difference frequency, and then you can identify regions with the highest frequency contrast. You can also do it with smaller marker numbers, although of course you still have some cost and labor saving, but then you will also lose some reliability because you have less markers in your case. And you can also do genome-wide association study, where you basically do statistical analysis between markers and traits, which typically utilize diverse population comprising differently related individuals. And this potentially has very high resolution, but then you also need a large number of markers which may not be available depending on the budget and population that you're using. You can also try genomic prediction, where you basically develop a computer algorithm to estimate the effect of each SNP to your trait. So once you supply it with the genotypic and phenotypic data, the computer algorithm will then try to estimate the effect of each SNP on your genome. And once you supply it with the genetic data, then the computer can output the predicted phenotypic data for your plants. And this can be very useful to select for like candidate parents, for example, for your gene population later on. And lastly, you can also use transcriptomics, obviously. This is part of the project that we are doing with Dr. Untung currently, where you try to find causal genes based on variation of gene expression levels of all the genes in the chromosomes and in the genome. And basically, you can try to find the gene expression differences just by counting the number of sequence data for the particular gene. So if the number of sequence data is high, then it's highly expressed, while it's not, then it's low. And then from there, you can correlate it to the trait that you are studying. And better still, if you already know the pathway that responsible for the, say for example, metabolite synthesis here, you can just look at the difference in expression levels in each of the enzymes in your pathway. And as you can see here, that only two part of the enzyme here has like very significant increase in expression. So you can tell that this enzyme here has have a very high impact on the production of the metabolites. You can also look at the sequence data to predict if there is any mutation in the protein that's involved in the pathway. So if you know that there is a mutation that disrupts the protein function, then you can say that this part here at least is not functional in this pathway. So that way you know that the cause is possibly because one of the genes is deactivated. And in closing, this is just barely scratching the surface, because there are also definitely many other techniques that we haven't tried yet. This is only some of the things we do because of the limitation of the budget and also equipment that we have in Indonesia. And in the future, I think since genetic regulation has many similarities to computer programming, so basically this is very amenable to AI analysis. So if you are interested in this kind of study, then I think AI will have a very high relevance in gene function prediction in the future. And that's from me. And if you have further question, you can contact me in this email. Thank you, Pak Yudis.
Thank you very much, Dr. Dani. Very interesting topic have been presented by Dr. Dani. Dr. Dani explained about the use genomic data. This is more downstream research, I think, in the explanation of the talk of Dr. Dani. And in genomic data, we can have we can do some activity like genetic resource management, and also we can do genetic analysis. And of course, the important thing from the genomic data, we can develop a marker for plant breeding to do the selection. I think it's also the important thing of the how to use the genomic data in the research. So thank you very much, Dr. Dani. Later on, we will discuss in the Q&A. So the last two speakers are from Indonesia, and the next two speakers from, of course, from abroad, from Lao PDR and from Malaysia. So I will call Dr. Khao Kam. He is a researcher from National Gene Bank Research Center of Lao PDR. So Dr. Khao Kam, you have 20 minutes for your presentation, and time is yours.
Hi, hello. Good afternoon. Can you hear me?
Yeah, very clear,
Nice,
your
guys.
voice.
Yeah, thank you very much, Mr. MC. And I would like to thank for organizers and to having me here to give opportunity to deliver my information from our gene bank or the Right Research Center to our meetings, important meeting. And also I would like to thanks for the previous two speakers. So I learned a lot from you, and especially from the breeding program and also from the molecular technique or biological technique, biotechnology technique in terms of the resource germplasm management. And so I should learn from you, like soybean core collections. So I have a large number of rice in my collection. So I would like to apply in such technique in my rice also. Anyway, so my name is Kukam. I work in the rice research centers, and especially in the plant genetic resources conservation and utilization unit, or we call it short name is the Gene Bank, under National Agriculture and Forestry Research Institute, or NAFRI. So, and under Ministry of Agriculture. So today, I would like to present very briefly about my gene bank collection, and also what achievement or what benefits from our collections in recent year. So, can I have my slide? Or do you allow me to open?
Yeah, it's
my
better.
screen?
Oh, okay.
Okay.
Yeah, the slide is there.
Thank you. Full screen, please. Yeah. Okay. Yes. So yeah, the topics I will present you today is rice germplasm collection and its contribution in Laos. So yeah, already I present briefly about my workplace. So my presentation today will cover introduction to Laos and the establishment of the gene bank, following by the current germplasm collection and also our achievement with the breedings. Next, please. Thank you. Yeah, some people, I think, yeah, of course, we are in the Asian country. Some people may not recognize where is Laos. Laos is the landlocked country. It's locked by 5 different countries like China, Vietnam, Cambodia, Thailand, and Myanmar. So the population is about 8 billion people. I don't know how many times compared to Indonesian populations. Okay. Yeah, my institute, since from the start, yeah. At the top is a map, Ministry of Agriculture and Forestry, state, and the NAFRI, National Agriculture and Forestry Research Institute, and the other institute, there are about 13 research centers. So my center is Rice and Cash Crop Research Center. Under center is the Lao Gene Banks over there. Yeah. So yeah, next please. So why we have gene bank? So in our culture, rice is the staple food, and also our people is more than 80% a farmer, and so of course we have a lot of rice land rest in our country, cultivated for long time. Also, rice is the one of the 5 priority crops for agriculture strategy development in our country, and also one of the strategy for agriculture development is the conservation and use of the genetic resources. So that's why we have started the collection, the rise from 1993, and then 1995, there was a gene bank exhibit in our center that you can see in Vientiane, capital city, at the first building, at the old building, and the right photo is the second building, another building of the gene bank. Those mean there are two gene bank building in our place, and 25 kilometers from the city center. Yeah. Next, please. Thank you. Yeah. For this building at the beginning, at the bit in 1995, as I already mentioned. So now, from that time until now, there are more than 14,000 acres of rice collected, and also including other vegetables and millets or something that you will see in the next slide. This. Yeah, and also the second building also is the another building. The new gene bank for us is at a bit in 2014, 10 years ago. So this building is now mainly is including two facilities, one cold room for medium-term storage, and another is long-term storage in the freezer. Next, please. Yeah. So this, as I mentioned before, so another facility in this building, we use long-term preservation under freezer at -18 degrees. Next, please. Yeah, now the total germplasm in our collection is, of course, mainly it's rice. It's almost 15,000 egg sections, and following by maize, beans, sorghum, eggplant, and vegetables. So in the vegetable is we keep in our main bank only for long-term preservation, and for the medium-term storage also in the sub-bank. Sub-bank means the center who responsible for the research on the horticulture crops, but also under NAFRI, under same institute, but different center. Yes, next, please. Yeah, so not only the seed bank, but we also have the field bank collection. For example, tuber crops. As you can see in the photo, some jam bean or sweet potato and some inside in this, but I'm not sure. And then if you are interested in, so I can present for the number of each section for us, but here I just like to show what we have it here. Thank you. Next, please. Yeah. So based on the collection, we have rice, vegetable, and other, of course. And I will focus on the rice breeding in our country. So of course, we are not only collect and conserve it, but we utilize the germplasm. So this is the demonstration plot of about 40 new released variety. So from 1995 until now, we have 40 modern variety released. Some of them are getting from using Palento live from the landrace or traditional variety in our collections. So yeah, next slide, please. So this is very short examples of some achievements from the breeding program used from our traditional or landrace germplasm that in our existing collections. So this new release variety, Sanakam 3. So this variety came from KDML 105, is the non-glutinate Thai aromatic rice. I think everyone knows it. And with the clause from the LG 3302. Is the LG 3302 is Lao traditional black rice, glutinate rice, and with good eating quality. And so then now we release the new variety with glutinate, with black color and also with HTML background. So these are some examples from our breeding. Actually, so many example released, so many example. Yes, but I'm sorry, I'm not good at preparation for on this sample. But of course, I promise if another series of meeting or webinar, I can show more and also include my colleague from the breedings unit and together with us to present you more if you are interested in. So yeah, for the next, maybe no more. Yeah, this is, I just to show some, recently we have screening for the deep plantings section, these LG numbers, these three samples from right side, either traditional or landrace in our collection. They can grow in the deep planting compared to the first from left control. So mean this germplasm will be useful for it will be used for paleontolite to develop a new variety for can be grow in the deep plantings. So for this, I think this is only two examples that I mentioned from the benefit from our collection. I have more. If I have a chance for the next time, I will deliver more of the example on this. So I think next please. Okay. That's all for my information. So if you have any question or to exchange to suggestion, I will try my best to answer. Thank you very much.
Okay, thank you very much, Dr. Halkam. Very interesting how to manage the rice germplasm in Laos and also how to utilize the germplasm become a breeding line. So I just want to explain to everybody that most of the Lao people, rice is a staple food, but Lao people is prefer to eat glutinous rice, right?
Yeah.
Rather than soft texture rice like in Indonesia. Then and also we have like TDK1 have been released in Indonesia. We call it as Impara Purwa, yeah. It is glutinous rice and it has adaptability to swampy environment.
Wow.
So thank you very much, Dr. Halkam. And the next speaker will be Ibu Rahiniza, yeah. Sorry, it's difficult to spell. Rahiniza. She is from Malaysian Agriculture Research and Development, MARDI. And I think Ibu Rihamiza will be deliver about the rice breeding program in Malaysia. You have 20 minutes, Ibu. Time is yours.
All right. Thank you, Dr. Yudhistira. Okay, I having problem with my camera. Can you give me a second? I cannot start my video. But let me share my screen first. Can you see my screen?
You can see your screen.
All right. So again, thank you, Dr. Yudhistira, for the kind introductions. Okay, Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon. To Dr. Puji Lestari, the chairman of BRIN, and my fellow researchers. Okay, this is a privilege to be here today to share some progress on research activities conducted in MARDI, specifically on the development of abiotic and biotic resistant rice varieties in Malaysia. Okay, So this is the outline of my presentation. I will cover the brief information of Malaysian rice industry. And then I will explain briefly on the development of rice variety. Rice variety development, tolerant to abiotic stresses. And then the second part is the development of rice variety resistant to abiotic stress. The previous part is specifically on drought, salinity, and submergence. And then for the abiotic stress, I'm focused on bacterial leaf blight disease. And then the conclusion. So for Malaysian rice industry, we have around 644,000 cultivated area, where only half of the figure is the parcel area, where we commonly plant the rice twice per year, 2 seasons per year. So our self-sufficiency level is quite low, only 63%. And then we are highly dependent on our neighboring country, such as Vietnam, to supply the white rice for us. And then we highly depend on specialty rice, such as basmati and jasmine from Thailand. Okay, and then as you can see, the granary average yield is around 4.42 ton per hectare, whereas the national average only 3.65. So there is a gap between these two figures. And then, so there is a lot of problem we need to tackle, especially on the infrastructure. All right. Okay, so next is how climate change affect Malaysia. So we face a flood problem during the heavy monsoon season, heavy rainy season. Okay, so we have the northern part is Mada. Mada is our main contributor of rice, followed by the IEDA Barat Laut Selangor. Okay, the below parts. So these two were highly affected by floods. And then also the drought happened in the east part of the Peninsula Malaysia. So for the saltwater intrusion, we rarely have this event in Peninsula Malaysia, but we received a report that it happened in Borneo. So maybe we can try our varieties to be planted over there. So in Peninsula, usually the saltwater intrusion only happened for the technical error, such as the failure of the water gate and also the damage of the water bund. Okay. So we focus on the development of rice variety tolerant to abiotic stresses, as I mentioned earlier. Focus on drought, salinity, and submergence. Okay, the first one is local drought tolerant rice variety development. So we did integration of drought tolerant gene or Q2Ls into illet variety. Okay, and then we screen in the trough in our rainout shelter. Okay, and then we further evaluated in field for yield evaluation and multi-location trial. So I'm sharing this screening method. So we impose the drought stress 30 days after transplanting, and then we maintain at 60 to 65 kilopascal, negative kilopascal. And then I will explain later. For 30 and 50 days, according to their treatment, we have mild and severe drought. And then lastly, we watering the plant to obtain the seeds, because if we are not watering, we cannot manage to obtain the seed, and we cannot evaluate in the further generation. So the rest are the standard data collection. Okay, this is what I mentioned earlier. The treatments. Okay, the mild reproductive stress and severe reproductive stress drought. Stress, we give 30 days after we plant, and then we draw the water out, and we left the soil to dry, and then we rewater only for 24 hours, only 1 day, rewatering 24 hours, only to allow plant to grain filling, to help the grain filling process, and then we harvest the line. Okay, and this is the examples of our identified variety or advanced line, MRT380. This is the photo the below photo is last week's. Okay, so the selection we made during the main season 2018-2019. Okay, the potential one, and then we further evaluated until the currently on off season 2024. So hopefully this variety can help our farmers to face the drought problem. Okay, our main problem is we cannot simulate the drought during our in our field trial. So what we did is we tried to build screening facilities for yield evaluation. Okay, in larger scale instead of planting in the trough. So this is in progress. Hopefully this facility will be successfully developed. Okay, and then the second part is development of salinity tolerant rice variety. So we integrate Salto QTL into our elite variety. And also we did the hybridization with mutant rice variety, Salinas. This variety we obtained from BSF FAO project, collaborated with Indonesia. Okay, as mentioned earlier by Dr. Puji. So that project bring more benefit to us in terms of germplasm exchange, specifically for this project. And then we did the screening and selection under salt stress at 6 decisiman per meter. Okay, because in Malaysia, we don't really have that high seawater intrusion. So we play around 6 decisiman per meter because rice can survive around 3 decisiman per meter square. And then we pick 6 according to iris method. Okay, and specifically at reproductive stage. Okay, for the seedling stage, we plan to use the seed treatment to allow the germination process, because in Malaysia, mostly farmers use broadcasting method instead of transplanting. The transplanting only applied on when the farmers is taking part in seed production system. Okay, because the labor cost in Malaysia is quite high, so they do the broadcasting technique. So the last part is evaluation of ASEAN rice net, and then MARDI advanced line. We did the screening, the same method. Okay, the ASEAN rice net is the project that we collaborated with IRI. I think other countries also participated in this project. So we evaluate the IRI's germplasm, the advanced lines, as well as our advanced line. So we'll plant it together. So I want to share our findings on the salt stress symptoms. So the spikelet sterility happened. This is they call it the peppery spikelet. Okay, the spikelet become white and cannot produce the seeds. Okay, this is finally will not contribute to the yield. All right. So and then we did the screening on the breeding line that we crossed with Salinas 2 with MR297, our early variety. 297 is the popular variety currently, and cultivated around more than 50% in cultivated area in Peninsular Malaysia. So we need to do something with this variety. We need to improve the other traits. For example, salinity and the other trait that we discussed today. Okay. So we also identified the MARDI advanced line. We identified 3, MR324, 346, and 347 that can withstand 6 decisiman per meter salt stress during reproductive stage. Okay, and then the last part for abiotic stress is submergence stress. I think Indonesia is more advanced on this. Okay, Malaysia I think University Putra Malaysia, if you ever heard about it. So they have a variety that have this gene, sub1 gene. So MARDI, we started also during the BSF FAO project. So currently the generation is quite advanced and under preliminary and advanced trial. So what we did is we screened by submerging the line in the pond as the picture below. Okay, and then we evaluate the yield, okay, for further selection. Okay, for the second part is for breeding for the disease resistant. Okay, as slides mentioned, the host resistant is the most efficient, environmental friendly method to cope with such diverse pathogen and pests. Why? Because Malaysia, we are very highly subsidized industry, means government spend a lot of money to help farmers. So what we did is we need a variety that can use less input, I mean lower costs, that have naturally resistant to any disease and pests. Okay. So I'm focused on bacterial leaf blight. We have others. For blast, we already solved the issue. And then currently we solve the sheath blight, the fungus issue. And then even though the sheath blight is quite problematic because we don't have any germplasm that naturally resistant to sheath blight. So what we did is we use a mutant line, we irradiate our elite variety. So I did not present it here because it is still early. Okay, maybe next time. Okay, so the other one is the newly emerged disease, which is bacterial panicle blight. Okay, this caused the loss of, I mean, hundred thousands of loss to our seed production because the bacterial panicle blight is seed-borne pathogen, so it will affect along the process, and then it caused the yield losses at the farmer's field. Okay, let's back to our topic, which is BLB. So this is the newspaper reported on the BLB. I think most of Indonesian can understand this. BLB kurangkan berat biji dan padi, okay. Reduce the yield. And then on the right side is you can see the farmer's field that affected the outbreaks of the BLB, I think during 2016. Okay. All right, what we did is we integrate the XA7 resistant genes into our elite variety. Why XA7? Because this gene is resistant towards our dominant Xanthomonas oryzae dominant patotype. Okay, we have other patotype, but we must tackle this one first. So I'm sharing the success story on the XA7 only. So okay, we improved the MR263 because this variety is very susceptible towards this disease. Okay, and with help of my colleague Encik Syaril, so he developed a functional marker to help me to select the potential lines. And as you can see, the dendrogram of resistant blue and susceptible red group in F6 population. So this functional marker can distinguish the resistant line, which is include the highly resistant, and moderately resistant. And then the red one is the susceptible one, include highly susceptible, and also moderately susceptible. So I'll discard the susceptible lines. Okay. And then we further evaluated until it fixed and advanced. Okay, so we identified 4 lines, advanced line, Y1831 until Y1834. Okay, and then we conduct a yield trial across 3 locations in Seberang Perai and Ara, which is the northern part of Peninsular Malaysia. And then the third one is in Mulong, the east part of Peninsular Malaysia. So what we did is we averaged the yield, and then we found that Y1831 is high yielding variety with BLB resistant trait as compared to the popular variety MR297, as I mentioned earlier, only about 5.76. But this variety currently is is further evaluated on multi-location trial for more location, about 10 to 8 locations across Peninsula Malaysia. Okay, to see its performance. Okay, and then the genotype by environmental analysis show that Y1831 is quite stable across these 3 locations. Okay, so this show that we successfully developed a high yielding variety with BLB resistant trait. Okay, I come to the conclusion. Is it quite early? Okay. Okay, we are developing actually. We are developing a climate resilient rice variety. Okay, for the first one is drought tolerant. Focus on reproductive stage only. We integrate the QDTY and gene related to deep rooting genes, and then currently in multi-location trial. And then the second part is salinity tolerant, also in reproductive stage. We integrate Salto QTL, and then currently in selection process, as well as the hybridization with mutant varieties, Salinas 2 we obtained from BSF FAO project, and also in selection process. The third one is Asenrasnet, as I explained, from Iris Germplasm or advanced line, and as well as Mardi advanced line, and currently in multi-location trial. Okay, the third one is submergent, focus on vegetative stage, which is one gene, currently in advanced trial. Okay, the second part is disease-resistant rice variety. I'm focused on bacterial leaf blight disease. We integrate XT7 gene and currently in multi-location trial. Okay, I think that's all. Thank you.
Thank you very much, Ibu Rahiniza from Mardi. Very interesting. Although Malaysia, mostly rice is imported from other countries, but the rice breeding program in Malaysia still continue, and in the pipeline, maybe there is some rice variety will be released by Mardi. And when I saw your presentation, I just realized now, because in our farmer, there is circulated rice varieties designated as MR. And when I asked to the farmer what is MR mean, they don't know. But where it's come from? They come from Malaysia. So I realized that MR is from Mardi, right? It's very interesting. Maybe yeah, it's just for information for you. Some of the rice from Malaysia also planted by our farmers. So, ladies and gentlemen, we have the speaker has been presented the presentation, and this time for answer and question session, and there is some question already written in the chat box, and I think some of the speaker also had been answered the question in the chat box, but for this session, I will ask for the audience to raise your question by raise your hand, or just through to the what do you call it? You can truly have a question in this session. Please, is there any want to raise question? If there is no, I will see in the chat box from Rifedi AM Damanik. I think it's already asked by it's already answered by Dr. Aris. From Novianto Fial Pagidung, for he asked to the Dr. Kwakam, but Dr. Kwakam is not yet answered this question. From Nurfianto Giar Pangidung, I want to ask to Dr. Kwakam, how is the development of red, brown, and black rice that is easy to progress in cooked rice in Laos, because in Indonesia, the majority of this type of rice, when into cooked rice, have a hard texture. Please answer the question. Or Pak Nufrianto, are you there? Maybe you can ask in the you can ask truly to the Dr. Kokom. Pak Nufrianto? Oh, okay. Dr. Kokom, please answer this question. Hello?
Hello, hi. Sorry.
Yeah.
Could you please excuse me with some noise?
Okay.
Yeah.
Nurfianto want to ask you about the development of red, brown, and black rice in your country. In your country, it must be easy to develop because majority the people of Laos is wanted this rice, preference to this rice. But in Indonesia, mostly those of rice variety red, brown, and black is very hard texture. So how about that? How to make the pigmented rice become more soft texture? Please answer that question.
Okay. Yeah. So actually, thank you for the very interesting question. So actually, my breeding colleague, the breeder, take care responsibility of this. So I'm so sorry about this. So I cannot answer directly, but please leave me your email address and then I will answer you.
Okay. Next
Yes,
question
so sorry.
from Bu Nurul Hidayatun.
Okay.
Hello, Kokam. You have 34 modern varieties from landrace as parental.
Yeah.
What is
Yeah.
the target trait in your breeding program? Usually landrace has long life, low productivity. How to overcome this problem?
Actually, mainly at the beginning, we focus on the high yielding variety. Yeah, from the starting point, but recently, of course, we get a little bit higher yielding, but we are targeting to the resilient rice variety, some of the biotic threat and the environmental threat tolerance varieties. Yeah, also the high yielding, of course.
Okay, is there any particular method for because if we closest with landrace, so mostly the recombinant will be very poor. It is high plant height, very tall, and lodgings and etc. So how to overcome this problem in your point of view?
Okay, yeah. My idea is, of course, we have to introduce other trees from the landrace by using the the good source breeding line from other, for example, from the Erie breeding line, and then we introduce some trees, the imported for us from the traditional variety. So then the background from the high yielding variety. So in that case, yeah, we have no problem about the use. Yeah.
Okay. Okay. Thank you very much for the answer.
Yeah. More questions. Yeah.
Yeah. I think some questions have been answered by the presenters. So some other maybe I would like to see from Nurfianto Giar Pangtingdung. The question addresses to Ms. Rahiniza. What are the current development and advancement related to KTL, genomic, and transcriptomic in rice resistant to lodging? Please, Mr. Rahiniza, answer that question.
All right, thank you for the questions. Okay, for the lodging resistant, we honestly we don't have the genomic approach to tackle this problem because we focus more on the phenotypic. So technically, the taller the plant, the higher the ability the plant to lodge during the heavy wind season. All right. So maybe there are other institutions in Malaysia who especially in university who are doing that studies, I guess.
Okay. Okay. Thank you very much for your answer. Dr. Danis already answered for Dr. Nurul Hidayatun question. From Fafi also. Currently, there are of silico to increase plant resistance to environment stress is trending to be studied. How is plant breeding for silico absorption, especially rice, a plant that absorbs large amount of silico? I think perhaps Dr. Aris, can you answer this question about the increasing of the role of silica to increase plant resistance in environment stress?
Thank you,
Aris?
Pak Yudis.
Yeah.
Thank you. Yeah. For the silica, actually, several years ago, we also have some research collaboration with one of private company who has like silica for as additional fertilizer, and yeah, they claim that it can improve rice resistance to like blast, and so for lodging. In our observation, yeah, actually it's quite potential, but maybe because of the price is quite high, and the application is not really, and farmers not really familiar for this. I think that it's hindered the application of this fertilizer, but I'm sure it has a potential for this, especially in rice for blast resistance. The use of silica, we found that it will increase the resistance of rice blast resistant by this.
Yeah. Thank you very much, Pak Aris. Actually, for species like rice, from the genus of Graminae, mostly this kind of crops can what do you call it? Can accumulate the silica. Mostly in Graminae type, silica is more abundant. But the problem is because our farmers is not to put the straw after they harvested in their field. They like what do you call it? Some farmer maybe they prefer to burn the straw, so that's why the silica content in the soil will be decreased. So I think cultural management also important to manage the silica status in the soil for what do you call it, for increasing the tolerance of the rice itself. So Pak Nufrianto has been, it's also has answered by Pak Aris. Is there any one of you to directly question instead in the chat box, please? Yeah, we have 5 minutes left, I think. So, Pak Aris, Jirkas, okay, Jirkas already. Yeah, I think there is no one question
Can
anymore.
I ask to Bu Rahiniza, Pak Yudis?
Okay, sure, say
Yeah,
sure,
yeah.
Pak Aris.
So,
Please.
Miss Rahiniza, thank you for your presentation. I just wonder, because we know that Malaysia, some part of Malaysia is in Borneo Island, and I wonder if you have like a swampy land which used for rice in that area, and how your breeding program or how your breeding program to tackle this area, especially for rice, maybe in your experience. Thank you.
Welcome. Okay, actually in Borneo, we have a different, authority. So there are two states over there, which is the first one is Sabah and another one is Sarawak. So for Sabah, they have their own research center for rice. Okay, and then for Sarawak, Mardi also help the farmers over there. Okay, for the swampy area, we just we have a Malaysian plan, which is the project based on the 5 years period. So only this time within 2021 to 2025, 2031 to 2025, we tackle on this salinity or swampy problem, salinity problem actually. So we just got the information that there are several areas affected by this salinity, especially for the swampy area. So we just discovered this information and we not yet conduct any trial over there. But we identified the three variety, the three lines that I mentioned earlier. So maybe for the next season or next 2 seasons, we will send the seeds to be planted over there. So that is our, we initiate the project by planting our potential tolerant lines. Yep. Yep. Indonesia is more advanced on the swampy land area. So I guess I can learn a lot from you all. Yeah.
Thank you. Thank you.
So we have about 10 minutes before we closing this session. Is there anyone to ask the question to the speakers? Pak Untung. Okay, Pak Dr. Untung Susanto. Kindly raise your question.
Yes, okay. Thank you very much, Pak Yudis, and thank you very much for all the presenters who had delivered for us so many knowledge, so many information for us. In this time, I just would like to ask to both from Laos and Malaysia. I'm wondering about the biofortification activity in Laos and Malaysia. Is there any biofortification breeding there? For example, for high iron and zinc content, and maybe for high vitamin A and other functional like glycemic index and so on. And for Laos, we saw that you have very nice color rice, yeah, black and red and so on. We have here Purwa, but it is a white rice, I think. So I'm wondering, and I'm interested so much for the color rice, and yeah, it is very useful for our health. And if Yudis, if possible, maybe we can also share some of the genotypes for us, and we can do exchange of the germplasm. I think there's two questions. I'm wondering about the biofortification activity in Laos and Malaysia, and maybe for Laos, can we exchange and share about the rice germplasm? Okay, thank you very much.
Thank you very much, Pak Untung. Please, Dr. Kukap, can you answer the question from Pak Untung? Dr. Kukap, are you there? If Dr. Kokang is left, I think he's left the room. So Ibu Rahiniza, please answer the question from Dr. Untuk.
All right. Thank you, Doctor, for the questions. Okay, for the Malaysia, the fortification, we focus more on the colored rice. So currently we have two varieties for red rice variety, colored rice with the red color. So initially we focus this variety more on Sarawak, which is the Borneo part, because in our Peninsula Malaysia, they don't prefer this type of rice. Okay, but we cannot manage to penetrate the market in Sarawak because they still prefer their local varieties, which is the traditional variety, which is they prefer the taste of the variety. Even though our variety have more antioxidant, okay, but the taste is quite different. So I guess we need to explore more on the local, the land races over there, and then we breed the one that's similar. Maybe we can improve that variety and help in terms of the yield instead of fortify the vitamin or the antioxidant. Okay. Maybe if you're interested, you can approach us to obtain the two colored rice. We also have the black glutinous rice. But in Malaysia, as I mentioned earlier, that our SSL is quite low, only 63. And we don't get any, I mean, the farmers is not interested to plant the specialty rice because they need to separately milling the rice and then they need to sow the rice, separately. Okay, so there is a lot of effort need to be done. And then they decide to plant the white rice. Okay, and then they go with the standard process. So yep, in Malaysia, the rice industry is quite challenging, and I got the information that if they don't get the subsidy, they will plant other crop. I mean, yep.
Okay.
Yeah, that's all.
That's the problem. Okay. The last question for Ibu Rahiniza, question from Triyasita Resmi. Do you develop varieties tolerant more than one trait? For example, drought and salinity and drought, or three kind of trait, drought, salinity, and submerged perhaps in one varieties. Please explain. I'm sorry, Rahiniza, please unmute.
Oh,
your...
I'm sorry. Okay, thank you for the questions. Okay, this is very good question. And honestly, we just started these activities by combining all the traits as I presented earlier, because we faced the problem to do the screening. We just set up the screening protocol and the screening facility. So after this, when we combine all these three traits or more than that, we have all the facilities that we can conduct the trial. Yep.
Okay, thank you very much. So I think time is over, but before I closing this session, I would like to hear from the presenter the closing statement. Please, Ibu Rahiniza first.
All right, so thank you so much for the opportunity given by the BRIN, especially thanks for Dr. Puji Lestari for being so nice to me. Okay, so hopefully we can further our collaboration for the next project. So I'll be grateful to be involved in any project that BRIN's propose. Yep, looking for our next collaboration. Yeah.
Okay, thank you so much. Please, Pak Dani.
Yeah, I also like to thank the organizer for inviting me to Pak Kapus and moderating this discussion, and also Bu Puji. And I'm also very happy to see Rahiniza again. We had a previous collaboration with Berger during the CodeF project, so it's nice to see her again after a few years, I think. So if there is also other collaboration in the future, I also definitely welcome it. And you can contact me in my email address that I provide in the end of my slides. Nice, thank you.
Last, Pak Aris.
Thank you, Pak Yudis. So yeah, I think similar to other speakers, first of all, we would like to thank ORPP for inviting me. And then I think this chance for us, I mean with Mahdi, with Lau researcher to build up our collaboration in rice breeding. And probably if there is any opportunity for material exchange, for genetics material exchange, we will very happy if we can get some material from Malaysia or Laos to enrich our genetic resources in our breeding program. I think that's all. Thank you, Pak Yudis.
Okay, thank you very much for speakers in this session. So because the time is limited, so we have to close it in 50 p.m. So I will not conclude that in this session. So but the important thing in this session, not only we have sharing some experience in related to rice breeding in each country, but also the most important thing is we can have collaboration in the next after this meeting, right? Like germplasm exchange and other thing to collaborate to increase the capability of each researchers, and also to build the strong collaboration between each country. Thank you very much for the speakers, and also thank you very much to the audience who still in the this session, still watching and still looking for the information of this in this session. And I will give applause to the 4 speakers, and I will give the session to the MC, the MC. Yeah, Bu siapa? Lupa lagi. Yeah, please.
Okay, thank you. Thank you, Dr. Yudhistira, for leading a very interactive discussion, and to all speakers for delivered a very interesting presentation. And as a token of our appreciation for sharing their valuable knowledge, we will give a certificate for the speakers and moderator to Aris Hairmansyh, SPMSI, PhD.
Thank you.
To Dr. Dani Satyawan, MS. To Kokam Filei Huang, PhD. And to Rahiniza Kamaruzaman, MSc. And to Dr. Yudhistira Nugraha, SPMP, as a moderator. Thank you for sharing your valuable knowledge. Ladies and gentlemen, this is the end of our agenda for today. I'm Andalusi Arini. On behalf of the committee, I would like to say thank you very much to our speakers, moderator, and all participants. See you in the next event. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very much.
Thank you very much.
Waalaikumsalam.
Thank you. Thank you.
Okay.